Hey guys, Miss Cote here, and welcome to this episode on viruses. I know once upon a time you learned or were supposed to learn about viruses in your biology class. So, for the sake of time, I'll not be spending any class time discussing this material. Everything you need to know about viruses will be in this video, and I will try and incorporate our new friend COVID-19 where I can. So, let's get started. First off, viruses are not considered to be a living thing. We learned earlier in the year that the smallest thing that can live is a cell. A virus is even smaller than a cell. It's basically made up of some genetic material and a protein coat. And because it's missing very important structures such as ribosomes, it cannot make its own proteins, among other activities needed for life, including reproducing. So when everyone talks about killing a virus, that's not technically true. You can't kill something that's not alive. A better term would be to destroy the virus. Therefore, viruses are parasites. They need to infect a host cell and use that cell's organelles to be able to reproduce and spread to other cells. First, let's take a look at the structure of a virus. Then we'll look at the two different ways viruses can replicate. So I said earlier that all viruses have genetic material. This genetic material, or genome, can be either DNA or RNA. I know you learned that DNA is double-stranded and RNA is single-stranded, but for viruses it doesn't seem to matter. The genome can be double or single-stranded, whether it is DNA or RNA. This is the main way that viruses can be categorized into families, by how their genome is organized. And depending on how many proteins or enzymes they need, there may be a few genes or many genes to code for those proteins. As far as the protein coat is concerned, this is a structure called a capsid, and its function is to enclose and protect the DNA or RNA. A capsid can exist in various shapes. For example, this is an example of a helical capsid, while this shape is called icosahedral. Viruses that infect animals may also have an additional outer layer called an envelope. This envelope comes from the cell membrane of the host cell. The virus takes pieces of it when it exits the host cell. The molecules within the envelope are a combination of the virus and the host cell. And we've seen this last guy before. In case you don't recognize it, this is a bacteriophage or phage. These were used in the Hershey and Chase experiments to determine that DNA is the genetic material. Bacteriophages are viruses that infect bacteria, and they have the most complex capsid known. It looks like a little spaceship with the DNA in the head of the capsid. The phage lands on a bacterial cell using the tail fibers as legs and injects the DNA into the bacteria through the tail sheath. And that's basically all you need to know about viral structure. Earlier I said that viruses were parasites. More specifically, they're called obligate intracellular parasites, which basically means that they can only reproduce inside a host cell. Also, viruses have a range of cells that they can infect. Some viruses can infect multiple species, while others are restricted to just one species. Some viruses can only infect a certain type of cell in one particular species. Talk about specializing. Here are some basic features of the replication cycle of viruses. First, the virus has to enter a host cell. There's usually some type of recognition system, like a lock and key fit between the virus and the host cell. When the virus enters the cell, the capsid comes off and the DNA is released into the cell. Once the DNA is inside the cell, it uses the cell's enzymes to make copies of its DNA. You know, that DNA replication process from the beginning of this unit. The DNA can also be transcribed into messenger RNA and then translated into the proteins that the virus needs, whether they are the capsids for the new viruses or the enzymes that the virus needs. Again, the virus makes use of the host cell's enzymes, ribosomes, transfer RNAs, amino acids, ATP, and other molecules to get this done. Then the parts are able to come together to form a new army of viruses, and then they exit the host cell, usually destroying it in the process. This damage leads to the symptoms that make you sick. I know I keep going back to bacteriophages, but since they are used quite often in research, these are the best understood of all the viruses. Working with and studying bacteriophages, scientists have learned that there are two possible mechanisms of how viruses can reproduce inside a cell. The two mechanisms are the lytic cycle and the lysogenic cycle. So let's start with the lytic cycle. The simplest of the two cycles, the lytic cycle usually ends with the death of the host cell. Phages who use this method to reproduce are often referred to as virulent phages, virulent meaning harmful in its effects. And I would think the death of the cell would be considered harmful. Similar to the previous diagram, first the phage needs to attach to the host cell. Then the phage will inject its DNA into the host cell and goes to work. One of the first proteins that's made is an enzyme that breaks the host cell's DNA into pieces. The viral DNA will make copies of its DNA and make copies of the capsid using the host cell's machinery. Self-assembly will occur, and then a new army of viruses will burst out of the cell, releasing them to go and infect other bacterial cells. This entire cycle can take as little as 20 to 30 minutes. That's pretty crazy. But bacteria have evolved defenses to fight off these attacks by the phages. Some bacteria have mutated surface proteins that are no longer recognized. If that doesn't work and the viral DNA gets inside the cell, some bacteria have special enzymes that recognize that it doesn't belong to the bacteria, that it's foreign, and these enzymes will chop up the viral DNA. Finally, some bacteria have learned to coexist with the phages, which brings us to our second replication cycle, which is called the lysogenic cycle. In the lysogenic cycle, the virus attaches to the host and injects its DNA. But instead of immediately making copies of itself and destroying the host cell, the virus's DNA is taken up into the DNA of the host. The incorporated viral DNA is now referred to as a prophage. At this point, the viral DNA is not doing anything, just hanging out in the cell. And every time the cell divides, so does the prophage. So basically, the virus is being sneaky and spreading itself through the new cells without actually making new viruses and infecting cells from the outside. The prophage can stay in this form until some type of trigger occurs that switches it to the lytic cycle. Then it would start making copies of new viruses that will eventually destroy the cell. Phages that use both the lysogenic and lytic cycles are called temperate phages. Obviously, these phages do not have the capability to infect our cells, but we can use the knowledge gained from them to determine how viruses can affect us. One of the biggest factors on how viruses replicate in animal cells is its genome. Not only if it is a DNA or RNA virus, but also if it's single or double-stranded. This figure shows how animal viruses are classified based on their genome. We have double-stranded DNA viruses, single-stranded DNA viruses, and double-stranded RNA viruses. Notice that single-stranded RNA is further separated into 3 different families. One type serves as the actual messenger RNA to use directly for translation, and this is where coronaviruses such as COVID-19 is classified. One type serves as a template for transcription, and the last one is a special group called retroviruses, which is used as a template to go backwards and make DNA. More on retroviruses in a minute. In addition to the genome, many animal viruses have an envelope surrounding the capsid as well. This envelope usually comes from the cell membrane of the host cell. When the viruses are leaving the host cell, they can take a piece of the cell membrane with them. These envelopes also have glycoproteins embedded in them that aid in recognition of other host cells for the virus. But as always, there are exceptions to the rule. Sometimes envelopes come from the nuclear membrane or even the Golgi apparatus. So to modify our earlier diagram with an enveloped RNA virus, such as COVID-19, these glycoproteins are used to bind to receptors on the host cell. This allows the virus to enter the cell. The capsid is digested, which releases the single-stranded RNA into the cell. Since this particular RNA is used as the template, let's see how that works. The viral RNA is transcribed to make a messenger RNA. This is done by a viral RNA polymerase because our cells don't go from RNA to RNA, so we don't have that particular enzyme. So the virus has to supply its own enzyme for that. Over on the left, the messenger RNA is translated to make glycoproteins for the cell membrane, which will become the new envelopes and capsids for the new viruses. Over on the right, the same viral RNA polymerase makes new copies of the viral RNA for the new viruses. After spontaneous self-assembly, the new viruses bud from the host cell, taking pieces of the cell membrane with the glycoproteins as its new envelope. Finally, one of the coolest and most complicated types of viruses out there are the retroviruses. These viruses are single-stranded RNA viruses that are capable of synthesizing DNA. Remember, the normal flow of genetic information is DNA to RNA to protein. So retroviruses can go backwards against the flow from RNA to DNA. And they can do this by a special viral enzyme called reverse transcriptase. The most famous retrovirus is the HIV virus. After the virus enters the host cell, it releases the RNA and enzymes. Reverse transcriptase will make double-stranded DNA from the RNA. The DNA will then enter the nucleus of the cell and incorporate itself into the host cell's DNA. The viral DNA is now called a provirus. From there, the cell's RNA polymerase can transcribe the proviral genes into RNA to either be used for the new virus genome, or to make more capsids, reverse transcriptases, and glycoproteins for the new viruses. And after assembling themselves into the little army of viruses, they bud from the host cell, destroying the cell in the process. Just a heads up, College Board has always liked retroviruses and reverse transcriptase. So just to quickly mention what's been happening with this new coronavirus called COVID-19. Our new virus is part of a large family of viruses that includes the simple common cold, SARS, which was first reported in Asia, and MERS, which was first reported in the Middle East. It's not very common that these viruses make the jump from animals to humans, and in these cases, SARS, MERS, and COVID-19, the virus originated in bats. These viruses specifically target lung cells in the respiratory system, with symptoms ranging from fever, coughing, and shortness of breath to more serious illnesses such as pneumonia, which can lead to death. And with any pathogen, prevention is key, especially now that the virus has made its way to New England. You can take care of yourself by staying away from sick people. Don't share your food and drinks with others. Don't touch your face, because that's where the virus will get inside you through your eyes, nose, and mouth. And wash your hands with soap and water. Soap dissolves lipids, which is what the envelope is made of. So you will expose the inside components of the virus, making it easier to destroy. And that's what you need to know about viruses. For the most part, this should be a review from what you should have learned in Honors Bio, with a few more details thrown in, since this is AP after all. So until I see you next, be a good human.